How do viruses replicate in microbiology? Imagine a tiny invader that cannot grow or reproduce on its own. Instead, it sneaks into a living cell and uses that cell's machinery to make more copies of itself. That's how viruses replicate. They don't have their own enzymes or organelles, so they rely completely on the host cell they infect. The process starts when the virus finds the right cell to infect. It does this by attaching to specific molecules on the cell surface called receptors. These receptors are like lock and key mechanisms. Different viruses target different receptors, which is why some infect only certain tissues or species. For example, the human immunodeficiency virus binds to CD for receptors on immune cells. Once attached, the virus enters the cell. Enveloped viruses, which have a lipid envelope, fuse their envelope with the cell membrane, releasing the viral core inside. Non-enveloped viruses, which lack this envelope, usually enter through a process called receptor-mediated endocytosis. The cell engulfs the virus into a vesicle, bringing it inside. Next, the viral capsid, which is the protein shell protecting the genetic material, is removed in a step called uncoating. This releases the viral genome into the cell's cytoplasm or nucleus, depending on the virus type. Now, the virus begins to replicate its genetic material and produce viral proteins. DNA viruses generally replicate in the nucleus using the host's DNA polymerases, while RNA viruses replicate in the cytoplasm with their own RNA-dependent RNA polymerases. Retroviruses, like HIV, reverse transcribe their RNA into DNA and then integrate into the host genome. During this phase, the host cell is tricked into making viral components. The viral genetic material is copied, and viral messenger RNA is translated into structural proteins like capsid and envelope proteins. The virus also produces enzymes needed for assembly and releases. Sometimes, the virus produces proteins that shut down the host cell's normal functions to prioritize viral production. Once enough viral components are made, they are assembled into new virus particles in a process called maturation. This can happen in the nucleus or cytoplasm depending on the virus. The final step is releases, where mature viruses exit the cell. Non-enveloped viruses often cause the cell to burst open, killing it. Enveloped viruses usually leave by budding, taking a piece of the cell membrane from their envelope. The entire cycle can take from a few hours to several days, depending on the virus and host cell. For example, bacteriophages, which infect bacteria, can replicate in less than an hour. In contrast, viruses infecting human cells might take 18 to 20 hours because of the complexity of human cellular machinery. Understanding how viruses replicate helps scientists develop medicines that block specific steps, like entry inhibitors that prevent attachment, or drugs that stop viral enzymes from copying genetic material. It also guides vaccine development by targeting key viral proteins involved in the cycle. So, in simple terms, viruses hijack host cells step by step, attaching, entering, uncoating, copying their genetic material, assembling new viruses, and finally leaving to infect more cells. It's a clever, if destructive, process that keeps viruses spreading.